Hi, sequence alignment is one of the powerful methods used in bioinformatics for pattern recognition as well as for prediction. Sequence alignment is actually looking for similarities between different sequences. It is a process of comparing and detecting similarities between biological sequences. These biological sequences can be a DNA sequence, RNA sequence, or the protein sequence. By identifying the similarities between sequence, there might be a relationship in functional, structural, or evolutionary aspects of different sequences. In sequence alignment, we are looking for common character pattern and to establish a residue to residue correspondence among biological sequences. So, before going into the details of sequence alignment, I would like to introduce few terms which is commonly used in sequence alignment. First one is alignment score. If you are taking two different sequences, maybe two different DNA sequences from two different organisms, let's say we are taking the hemoglobin genes from human being as well as from the horse. If you are taking two different hemoglobin genes and you are comparing, and the percentage of similarity or the extent of similarity can be expressed as alignment score. If two different sequences, DNA sequences are very similar, the alignment score will be high. If they are not that much similar, the alignment score will be less. Depending on the program we are using for the sequence alignment, the scoring pattern will be different. So if the similarity is more, the alignment score will be high. If the similarity is less, the alignment score will be less. Distance between the sequence is actually the reciprocal of the alignment score. So how much distant they are, how much dissimilar they are, that's what called the distance between the sequence. Then the Hamming distance means if between two different sequences, if you see this example, there are three dissimilarities or substitutions happen in this sequence. So the Hamming distance is the actually the non-matching characters present in the sequence. This sequence can be DNA sequence, RNA sequence, or a protein sequence. So if the Hamming sequence distance is high, the distance between sequence will be also high. Then the alignment score will be less. So alignment score is actually telling how much similar the sequences are. Distance between sequence is actually how dissimilar is the sequences are. The sequence alignment is doing this way. We are writing two different sequences and we are looking for the similarity. But in terms of evolution, there are different types of evolutionary forces acting on during the evolution, though that includes substitution, insertion, and deletions. But the chances of substitution is way much higher than insertion and or deletion. So when we are doing the alignment, what we are trying to do is to minimize the number of substitution, insertion, and deletion between two sequences. So we want to fit in both sequences by introducing gaps and all. So that process is called edit distance. So after the editing, like after the aligning the two sequences together, we are looking for what is the alignment score and what is the distance between sequences. So there is a fitting mechanism happening in the sequencing method. So this is the possibilities. The match, when the two residues are same, it can be called a match. And the mismatch is when two sequences are different. That can be due to a substitution. And finally, we have a gap. This gap can be either due to a deletion in this sequence or due to an insertion in the other sequence. But in evolutionary point of view, the chances for the substitution is way much higher than the chances for insertion or deletion. Here, the similarity score is calculated by using this equation, but depending on the type of program we are using, this calculation may be different, the scoring pattern may be different. So the variation between two sequences are due to substitution, insertion, and deletions, and the similarity between two sequences is actually a measure of evolutionary relatedness of two different sequences. If two different sequences are very much related, that means they are evolutionarily related. Yeah, just I said before, the hemoglobin genes from the humans and horse are very much related because they are evolutionarily related. They are originated from the same ancestral protein. Then the sequence homology means if two sequences are originated from the same ancestral sequence, sequences, we call these two sequences are homologous in nature, just like we are telling in the evolution. Sequence identity means when two sequences are exactly same. That's called a sequence identity. If in a place of time in one sequence they have in a particular position t and the other one is also having it t. If one is having a and the other is also having a, if they are having exact nucleotides or exact amino acids, we call it as sequence identity. But next thing is sequence similarity. Sequence similarity means they are dissimilarity between two different sequences, but it doesn't really affect the protein. Suppose if in one position a protein is having a positively charged amino acid, but in the other one a mutation happens and it is changed into another positively charged amino acid. So one positively charged amino acid is changed to another positively charged amino acid. These two amino acids have a similar characteristics. So we can say that like they are similar, not identical. So that's what we called sequence similarity. So there is a difference between identity and similarity. In case of identity, they are exactly the same, but in case of similarity, they are not the same, but they are physiologically similar. Maybe one amino acid is replaced by physiologically similar property, similar amino acid. Maybe a hydrophobic amino acid is replaced by same similar size amino other hydrophobic amino acid. So that's the difference between identity and similarity. So one another like feature of sequence alignment is that if the length of the sequence is less, the chances of getting similarity between two different sequences is high. So if the shorter the sequence, higher the chance of alignment. And if the sequence is larger, length is high, the chances of random chance of getting alignment is less. So based on the, there is a relationship between the like accuracy of the alignment and the length of the sequence. And this is a graph representing the sequence identity and the sequence length. Here they have divided it into three different zones. Midnight zone, it means that if a sequence similarity, like if a sequence of length 150 has got a similarity of 20 percentage, that means it is still in the midnight zone. We cannot actually take the data because it may be due to the random chance. So here we have twilight zone. The twilight zone, we are not sure whether it is a true similarity or the similarity due to the random chance. Okay, so if it is smaller the sequence and if it is fallen the twilight zone, we need to do some more experiment to make sure that this one is not due to the random chance. In the safe zone, if a sequence of length 150, it got a similarity of 60 percentage, it's in the safe zone. That means we can surely say that these two similar sequences are similar. Okay, so if we take a one example, if we have a sequence of 100 base pairs, if it two different sequences of 100 base pairs, if it has a similarity of 20 percentage, we cannot believe the data. The data is due to the random chance. If it is have a 25% similarity, it's in a twilight zone. We are not sure whether it is a true similarity or due to the random chance. If it has a similarity of 50 percentage, we can surely say that this similarity is due to the evolutionary relatedness between the sequence. So the point is that if the length of the DNA sequence is less, there is chances that the dissimilar sequences show similarity just because of the random chance. So there is a threshold based on the sequence length to identify whether they are showing a true similarity or due to the random chance. Methods of alignment. There are two different methods of alignment based on the number of sequences are comparing at a time. One is the pairwise alignment, another is the multiple sequence alignment. In the pairwise alignment, we are comparing two different sequences at a time. It can be a DNA sequence, RNA sequence, or a protein sequence, but we are comparing only two different sequences at a time. In multiple pair sequence alignment, we are comparing three or more sequences to see whether how much similar they are. So that's the difference. How much sequences are compared at a time. So pairwise alignment, it is used to find best matching piecewise or alignment between two different sequences, and there are two different types of pairwise alignment. It can be a local alignment or global alignment. In this figure you can see that in the local alignment, the first figure, we are actually trying to see how much similarity they are showing in a localized manner. So the first sequence is bigger and second one is smaller, and we are trying to fit in fit in two different sequences best local way. But in the global alignment, we consider that both these sequences are in similar length. So we are trying to find out end-to-end comparison between these two sequences. Here you can see there is no, in the local alignment we do not actually introduce any gaps, but here we have introduced gaps to make sure that both of them are in same length. And sometimes it is better to do the local alignment, and sometimes the global alignment is better. So if I want to give few more details about that, the local alignment do not assumes that two different sequences are in similar length. It is of different length. So one is bigger, another is smaller, and we want to find the best local fit between these two sequences. And it finds only local regions of highest similarity, and it is used to align divergent sequences. If two different two sequences are distantly related, it is better to do a local alignment than the global alignment. If it is a very similar sequences, it's better to do the global alignment, not the local alignment. So uh, it can compare sequences of two different lengths. The global alignment, sorry, we are looking for an end-to-end alignment, and if two sequences are very much similar, it's better to do the global alignment. So we are checking for the similarity in the entire length of the sequences. It aims to find the best possible alignment across the entire length. So it's more applicable to closely related sequences. It fails to recognize high level of local similarity between the two sequences, because any sequence we are getting, we are trying to make them in the same length. And there is a combination of two, that means it's semi-global or global alignment. It's a hybrid method. It is actually used to find the alignment in such a way that only it's actually looking for the global alignments, but the gaps are allowed at the beginning and or the end of the sequences. We cannot actually put the gap in between. We can either put the gap in one end or the both ends. So it attempts to find best fitting between two different sequences, especially one with the overlapping genes, overlapping areas of similarity. So if we are suspecting that two of our sequences having a similar region or overlapping region of similarity, it's better to do the semi-global or global alignment. Thank you.